Okay, now we will see the strategies for enhancement in food production. As we all know, the world population is increasing, so there will be a demand in food production also. So there is a need to increase the food production. So from both, where will be increasing it? From both animals and plants only. Okay, and the first one we gonna see is animal husbandry. What is animal husbandry? What is it? It is an agriculture practice of breeding and raising of livestock, is called as animal husbandry. Okay, and it deals with the care and breeding of animals like buffaloes, cows, pigs and then horses, cattle, sheep, goat, etc. by human for various products from them, like what? Milk, meat, wool, everything. Okay, and then poultry farming and fisheries are also considered as a part of animal husbandry, and about 70% of the world's livestock are from India and China only, 70%. Now let us see the management of farm and farmhouses. That is, one is dairy management, another is poultry farm management. Dairy means it mainly deals with is the process and the system to improve the quality and the quantity of the milk. Okay, milk yields mainly depends on the quality of the breed. Milk actually depends upon the type of the breed only. And the dairy farm management should be, there are many processes while selecting the dairy management. That is, first is selection of the good breeds with potential of high yields, that is which breed yields high milk, and then also cattle should be housed well and then maintained and should be kept in a disease free condition, and also there should be a regular inspection and keeping proper records of all the activities of the farm, and then there should be also a regular visit to the veterinary, and then cleanliness and hygiene for both the cattle and handle are very important during milking, storage and transport of the milk and its products. What is poultry? What is that? It means rearing of domesticated birds. Okay, domesticated birds for food and their eggs, like example ducks, chicken, geese, turkeys, etc. And there are also some management which includes in poultry farming is first selection of the breed only, and then housing should be safe and proper ventilated, and proper sanitation should be made, and then regular visit to the veterinary is also main one only, and then maintenance of healthcare and hygiene of the poultry. Okay, now let us see about the bird flu. Bird flu is otherwise known as avian influenza or HPAIH5. It is the illness caused due to the birds only. It the viral disease that affects the wild birds and then affects the capitated wild birds and domestic poultry also. And there are two forms of avian influenza. One is low pathogenicity and another is high pathogenicity. Low means, what is low? Low pathogenicity means nothing much, they are mostly harmless, and infected chickens will react with general flu like symptoms only in low pathogenicity, but whereas in high they are extremely contagious and kills the approximate 90% of all the chicken present in the poultry. Okay, how to prevent a bird flu? Like cooking poultry products thoroughly above 70 degrees Celsius, and then keeping our hands clean and hygiene, avoid visiting the poultry markets oftenly, and then avoid contact with the infected poultry, and last, seek medical advices whenever it is needed. Okay, what is a breeding? What is breeding? Yes? Yeah, that is correct. Interaction between the two animals which have a similar in most characteristics, general appearances, features, size, etc. And the animal breeding is of three types, three categories. One is inbreeding, outbreeding and controlled breeding. Let us see about inbreeding now. Inbreeding means mating of the relatively close animals within the same breed of 4 to 6 generation in common. That is called as inbreeding. Okay, and what happens here is they will select the superior male and the superior female of the same breed and they will make us to interbreed, which results the progeny. And the progeny obtained from such type of mating are evaluated and then superior males and the superior females are selected from that. In cattle, how they select is more milk per lactation. Look, I mean more milk per location is described in the cattle, whereas in bull it is buffalo, it is superior female is one which gives superior progeny. And what are the advantages means inbreeding is done to evolve a pure line of breed. That is a one main advantage. And then it also increases the homozygosity. Homozygosity is nothing but the position of two identical alleles of a particular gene. And then the productivity of the inbreed population can be increased by selection at each step. And there are also some disadvantages which leads to inbreeding depression and thus reduces the fertility and even the productivity of the cattle or productivity of the cattle. And the next one is outbreeding. Outbreeding is nothing but the breeding of unrelated animals either of the same breed but they having not common ancestor between 4 to 6 generations. And this is also called as there are two types, right? One is three types: outcrossing, crossbreeding, outcrossing, crossbreeding, and then interspecific hybridization. Now let us see what is outbracting is. What they do is the practice of mating the animals within the same breed but having no common ancestors, that is 4 to 6 generation, and the offspring produced from that is called as a outcross. Okay? And whereas crossbreeding, what they do is the mating of the superior males of one breed and the superior females of the other breeds, and this is called as crossbreeding. Okay, why is done is to combine the desirable characters of two breeds into a single individuals. So this type of crossbreeding is done. And last one is interspecific hybridization. What they do is animals of one species is interbreeded with the other species, other kind of animal species. That example mule, an offspring which is from male donkey and a female horse. Okay, now let us see about the control breeding techniques, that of two types. One is artificial insemination and multiple ovulation embryo transfer technology. Now we will see each one by one. What they do in artificial insemination is they take the superior male and take the semen from it, and then what they do is they inject the semen into the reproductive tract of the female cow. Okay. But the success rate of this artificial insemination is very low, but they have some advantages. What happens is the semen collected can be used immediately or can be even stored and used for later use also. And then the semen from a desired breed can be easily transported in the frozen from a distant places where the female is available. And also it helps in the overcoming several problems in normal mating. Okay, and the next technique is multiple ovulation embryo transfer. What they do is, again this technique, they select a superior cow and they give the hormone which is known as follicular stimulating hormone. That what happens, the follicular maturation occurs and the super ovulation. And in normal cases there only one eggs will be produced, but in this case 6 to 8 eggs are produced at a time. And what happens in this time, by natural mating or like even by artificial insemination, they will inject the semen, and then in next stage it leads to fertilization and embryos at the 8 to 32 cell stages. What happens here, the embryos are recovered non-surgically, which they can genetic mother and the genetic mothers again ready for another ovulation, and the taken embryos are transferred to the surrogate mothers. This is the technique called multiple ovulation embryo transfer. Okay. Next we will see apiculture. What is apiculture? Honeybees. That includes the maintenance of the hives of the honeybees for the production of honeys and the beeswax. This procedure is known as apiculture. And the commonly used bee is Apis indica. It is an Indian bee. Okay, what is the economic importance of bees?
Honey.
Yeah, honey. It is a highly nutritive food product which is also used in medicinal products also. And it also used in the manufacturing of cosmetics and polishes of various kinds. And then honeybees are the pollinators for many crops, like example sunflower, apple, pear, etc. And another product of bee is used as a bee venom, which is used in the treatment of snake poison and arthritis, snake bite and arthritis. Okay. Okay, next is fisheries. Fisheries include rearing, catching and selling of fishes. And now let us see the economic importance of fishes. They are internationally traded commodity, and then about 40% of the global production enters international trade against 10% for the meat. And then what is the turnover? It is 102 billion in 2008 itself. And then exports of fish products exceeds those of meat, dairy, cereals, etc. that exist other than these products. And in many developing countries, foreign exchange from fish exports the finances other than food imports. And it also provides income and employment to millions of fishermen and farmers who are especially located in the coastal areas. And fishes also are used as a food as a good source of vitamins, proteins and rich in iodine. And fish liver oil is a natural source of vitamin A which can be used in medicines also. Okay, now let us see about plant breeding. We saw animal breeding, then what is plant breeding then? Yeah, whichever characters we like or we want to get in another plant. Okay. Plant breeding are used in various techniques like biotechnology, bioinformatics, genomics and then agriculture, horticulture, etc. Okay, now let us see the steps in plant breeding. The first one is selection of the variability. Okay, species and the relative crops are first selected for the variability, and the next step is evaluation and selection of the parents. What they do is, it is carried out by the evaluating germplasm. What is germplasm? What is germplasm? What happens is collection of plants having a diverse alleles for all the genes in a given crop, that is germplasm. What they do is, it is carried by evaluating all the characters of the same gene and then to identify the plants with desirable combination of characteristics. This is the second step. And the third step is cross hybridization among the selection of the plant parents. Whichever parent we selected will be what we will be doing, cross hybridization in it. And the next stage is selection and testing of superior recombinants. This step includes the selection of plants among the progeny hybrids with desired combination of the characters. And the next is testing, release and commercialization of new cultivars. Evaluation is done for the newly selected lines for their yield and the other agronomic trained for quality and disease resistance. Okay, these are the steps involved in the plant breeding. Okay, what is the first step? Tell me the steps.
Collection of variability.
Yes, and then?
Evaluation and selection of parents.
Yes, and then?
Cross hybridization among the selected parents.
Okay.
Selection and testing of superior recombinant.
Yes, and the last?
Testing, release and commercial of new cultivar.
Okay. In India, agriculture accounts about 33% of India's GDP and employs near 62% of the population. Okay, 32% is in India is agriculture only. Okay. Now let us see what is a green revolution. It refers to the large increase in crop production by using a modern techniques, agriculture techniques. Okay, who is the father of green revolution in India? Okay, it was Swaminathan who has been called as a father of green revolution in India for his role in introducing and further developing the high yielding varieties of wheat in India. He is the founder of Swaminathan Research Foundation. Okay. Now let us see some examples for this green revolution. What is that? Is sugar cane. We will take sugar cane. Sarcharum barbary, that is originally originated from North India, and Sarcharum officinarum of South India, which have a thick stem and a high sugar content level, but whereas in North India sugarcane, it doesn't have any high content. So what they did, they interbreeded both of them, and the hybrid variety came, okay, and which it can be grown in North India with a high sugar content, and this is one type of green revolution. Understood? Okay. Now let us see the plant breeding for disease resistance. These are the steps to be taken in the plant breeding for disease resistance. The first step is source for the resistant, identifying the source for the resistant, and then methods of breeding for the disease resistant and also testing the disease resistant. Last is selection for the resistant lice. These are the pathogens causing the disease in plants like bacteria, fungi, virus, phytoplasma, nematodes, etc. There are different types of plant disease also, like example crown gall, club root, fruit rot, canker disease, leaf crawling, and these are caused due to environmental host factors also. Okay, now let us see the advantages of plant breeding. The first one is it enhances the food production by reducing the losses due to disease. The first one, okay, and another, it reduces the dependence on the fungicides and the bactericides. Okay, and these are the methods for breeding for disease resistance. The first one is conventional method, that comes under hybridization selection. Another one is mutation method. First we will see conventional plant breeding. What happens here is involves crossing selected parent plants, okay, and then chosen because they have a desirable characters such as a high yield or a disease resistant. What happens, they select the parent plants because they have a desirable character, has a high yield, or they have a disease resistance. And then what they do is the breeder's skill lies in the selecting, okay, the breeder's skill lies in the selecting the best plants for the many and the varied offspring. These are grown and tested in a subsequent years, and this process called as artificial selection because people select which organisms get to reproduce. So it is called as artificial method. And whereas the mutation breed, there are two steps. One is choice of material and choice of mutagen. What they do is the best adopted variety of crop should be chosen for mutagenesis, and because such variety are acceptable agronomic features. First is choice of material, another is choice of mutagen. The choice of mutagen depends upon the plant parts to be treated. What they do is chemical mutagens are more preferred for seed treatment and radiation for the treatment of vegetative parts, but in the case of irradiation, the plant parts are immediately planted to raise the M1 plants from them. In case of chemical, its seeds are usually pre-stoked for a few hours to initiate, and the metabolic activities export to the desired mutagen, then washed in the running tape water to remove the mutagen present in them. Okay, now let us see about the plant breeding for developing insect pest resistant varieties, and like example morphological, physiological and biochemical characteristics are responsible of insect resistant in host crop plant, like example hairy leaves of the plant which results to the resistant to the pest. Okay. Now let us see the plant breeding for improved food quality. The first one is biofortification, which is divided into three types. One is genetic engineering, plant breeding and agronomic approaches, which includes mineral fertilizers, foliar fertilization and plant growth promoting microorganisms. Okay, there should be an improvement in the breeding of crop due to some reasons. The first one is lack of adequate food with nutritional value. Okay, that was the first cause, and then majority of peoples are unable to get their enough fruit, vegetables and fish and meat. So, they suffer from a hunger disease or a deficiencies. And then essential micronutrients are absent from the diet. So, which increases the risk of the disease, reduce the lifespan of the human and their mental health will be disturbed. These are the, that is why plant breeding, that food quality should be increased. Okay. Now, let us see about single cell protein. Single cell protein is nothing but bioconverting waste into food. Okay. Single cell protein is nothing but a supplement or an alternative source of protein that is not supplied by a traditional or conventional agriculture protein production. Okay, what happens here is microbes are being grown commercially as a source of single cell proteins. Like example, filamentous fungi, that is Fusarium graminearum, and then bacteria, that is Methylphilus methylotrophus, and then algae, that is cholera. Advantages of this single cell proteins include the microorganisms used for the production of single cell proteins are very fast growing and also produces a huge amount of proteins from very small area of land. And these microbes can be grown on anywhere like water, from potato or from processing plants, straw and then animal manure, sewage, etc. And this reduces the environmental pollution also. Okay. Next we will see about plant tissue culture. What plant tissue culture, what they do is the tissue sample is scraped from the parent plant. They'll take or cut the specific part of the plant from the parent plant, and then the tissue sample is placed in a agar growth medium containing nutrients and auxins. What happens next is some samples develop into tiny plantlets in the petri dish, and these plantlets planted are into the composite, which becomes a new plant. And these are the benefits of the tissue culture. What happens is in plant prone to virus diseases, virus free excipients can be cultivated to provide virus free plants, and then this plant tissue banks can be frozen then regenerated through a tissue culture. Plant culture is in approved media are easier to export than in the soil grown, whereas soil grown plants as a pathogen free and take up a little space. It also allows a fast selection of crop improvement, and then explants are chosen from superior plants and then cloned. And the next method is somatic hybridization. What they do is they allows a fusion of two different plants to obtain a new character or new hybrid plant with the both characteristics of the parent plants. It is carried out by the following steps. The first one is isolation of the protoplast from the suitable plants. They will select the parent plant and they will isolate the protoplast from the suitable plant, and then fusion of the protoplast of the desired species that they are fused, and then identification and selection of somatic hybrid cells, and culture of the hybrid cells and regeneration of the hybrid cells. Okay. Okay. And this is example for the somatic hybrid, that is it is a tomato which is produced by the fusion of tomato and potato protoplasts. It's called as tomato. Okay. Okay, after a break we will be seeing the next chapter.